Hello everyone, I am Dr. Trupti and welcome to my YouTube channel Enjoy Biochemistry. In the video lecture series on vitamins, today let's learn about vitamin B6, that is pyridoxine. In addition to that, we will also see one case study on pyridoxine deficiency. At the end of this session, students shall be able to describe vitamin B6 with respect to its chemistry, dietary sources, daily requirement, metabolism, various biochemical functions, deficiency manifestations, toxicity and its estimation. Vitamin B6 is a water-soluble vitamin and it is a collective term for various natural forms of vitamin B6. Those are pyridoxine having alcohol group, pyridoxal having aldehyde group and pyridoxamine having amino group, and all the forms of vitamin B6 they are derived from pyridine. So they are pyridine derivative. This is the structure of pyridine. This is structure of pyridoxine where at carbon number 4 this methyl alcohol group is attached, and all the forms of vitamin B6 they differ with the groups present on this carbon number 4. In the pyridoxamine, amino methyl group is attached. In pyridoxal, here aldehyde group is present, and this is pyridoxal phosphate PLP, and here phosphate group is linked here with the help of enzyme pyridoxal kinase, and ATP is also required for this formation of pyridoxal phosphate. This pyridoxal phosphate is the coenzyme form of vitamin B6. Vitamin B6 is derived from various plants as well as animal sources. Plant sources include wheat germs, cereals, green leafy vegetables, legumes, pulses, oil seeds, etc. And animal sources like egg, milk, meat, fish, they are the rich sources of vitamin B6. This can also be synthesized partly by intestinal bacterial flora. The requirement in adult is 1 to 2 mg per day, and in pregnancy and lactation it is 2.5 mg per day. Now let's see how this vitamin B6 is metabolized in the body. So it is absorbed, digested and absorbed in the proximal jejunum by passive diffusion when it is present in high concentration, and at low concentration it is absorbed by active transport. Once it is absorbed in the intestine, it is transported to liver where it is converted to its coenzyme form PLP, and then from the liver it is transported in the circulation bound to albumin, and it reaches the target organs or cells, and the pyridoxal phosphate cannot cross the cell membrane, so first it is dephosphorylated, and once it reaches inside the cell it again becomes phosphorylated, and it is stored in the form of pyridoxal phosphate in liver, brain, kidney and muscle. But 80% of the pyridoxal phosphate is stored in muscle linked with the glycogen phosphorylase, and it is finally excreted in the form of 4-pyridoxic acid. And in plasma, vitamin B6 is mainly present in the form of pyridoxal phosphate, that is 60% is pyridoxal phosphate, 15% vitamin B6 in plasma is pyridoxamine, and 14% is pyridoxal. Now coming to various biochemical functions of vitamin B6, and the functions are attributed to its coenzyme form, that is PLP, pyridoxal phosphate. It is the coenzyme of various enzymes involved in more than 100 biochemical reactions, but this PLP is mainly important in amino acid metabolism. However, it is also required for lipid metabolism, that is sphingolipid synthesis, as well as glycogen metabolism. So the first important function of PLP is in transamination reactions. Second, decarboxylation reactions. It is also required for non-oxidative deamination. Heme synthesis, it plays a very important role. It is required in tryptophan metabolism, glycine metabolism, methionine and cysteine metabolism, glycogen metabolism, that is in the carbohydrate metabolism, sphingolipid synthesis, that is lipid metabolism, and coenzyme A synthesis. So these are the various functions, important biochemical functions, where this pyridoxal phosphate is required as coenzyme of various enzymes. Let's first see the role of PLP in transamination reaction. What is transamination? There is transfer of amino group from amino acid to keto acid to form a new amino acid and a keto acid. And here the example is the amino group from alanine, it is transferred to alpha ketoglutarate to form pyruvate and glutamic acid. And this reaction is catalyzed by enzyme alanine transaminase, and this is the enzyme which require PLP, that is pyridoxal phosphate, as a coenzyme. The second transamination reaction is amino group from aspartate transferred to alpha ketoglutarate to form oxaloacetate and glutamic acid. This is carried out by the enzyme aspartate transaminase. This is called as AST, and this is the PLP dependent enzyme. So these are the important transamination reactions where the PLP plays very important role. Now in decarboxylation reactions also PLP is required. The first decarboxylation reaction is formation of histamine from histidine. So for histamine formation, PLP is needed. Second decarboxylation reaction involves formation of GABA. It is a neurotransmitter and it is formed by decarboxylation of glutamate. Third reaction where the PLP is required for decarboxylation in the tyrosine metabolism. So tyrosine is converted to DOPA. From DOPA we get dopamine by decarboxylation where PLP is required, and further from dopamine we can get norepinephrine and epinephrine. The fourth in the tryptophan metabolism, from tryptophan we can get 5-hydroxytryptophan, and then the decarboxylation of 5-hydroxytryptophan leads to formation of 5-hydroxytryptamine, which is also called as serotonin. So this PLP is required for histamine formation, for GABA formation, dopamine, norepinephrine, epinephrine and serotonin formation. So whenever there is deficiency of vitamin B6, it leads to decrease in all the neurotransmitters like GABA, epinephrine, norepinephrine, serotonin, and this is responsible for various neurological symptoms seen in vitamin B6 deficiency. PLP is also required for non-oxidative deamination of OH containing amino acids like serine. From serine by dehydratase we get pyruvate, and there is release of ammonia. Here this dehydratase is PLP dependent enzyme. From threonine we get alpha ketobutyrate and ammonia is released. This is also PLP dependent enzyme. So non-oxidative deamination requires PLP as coenzyme. PLP also plays a very important role in heme biosynthesis. The first step of heme synthesis occurs in mitochondria, and this step is succinyl-CoA plus glycine. There is formation of delta aminolevulinic acid, and this is catalyzed by enzyme delta aminolevulinic acid synthase, and this enzyme is a PLP dependent enzyme, and this is the first committed and rate limiting step of heme synthesis. So if there is deficiency of PLP, there will be decreased heme synthesis, and that's why in pyridoxine deficiency anemia is seen. Now let's see how it is involved in the tryptophan metabolism. Tryptophan metabolism, there is formation of 3-hydroxykynurenine from this tryptophan through various metabolic reactions. Now what happens to this 3-hydroxykynurenine? It is further metabolized to acetyl-CoA through the series of reactions, and one of the important step is formation of this 3-hydroxyantranilate from this 3-hydroxykynurenine, and this reaction requires kynureninase, the enzyme which is PLP dependent enzyme. Now what is the fate of this 3-hydroxyantranilate? The 3% it goes to formation of niacin. Niacin is vitamin B3 and the coenzyme of niacin is NAD. So for formation of these coenzymes and niacin, 3% of tryptophan it is channelized to form this niacin. And from the 60 mg of tryptophan we get 1 mg of niacin. So niacin can be synthesized in the body from amino acid tryptophan. Now what will happen if this PLP is deficient in case of vitamin B6 deficiency? So this 3-hydroxykynurenine cannot be converted to 3-hydroxyantranilate, which further cannot lead to formation of this niacin. So this step will be blocked, and the niacin level will be decreased. So here in case of pyridoxine deficiency, there will be deficiency of niacin also. And what will happen to this 3-hydroxykynurenine, which is now not able to be converted into 3-hydroxyethylate? The levels will increase and it will be channeled to another reactions like formation of xanthurenate, and this xanthurenate will be excreted in the urine. So in case of vitamin B6 deficiency, there is deficiency of pyridoxal phosphate, and that's why this enzyme kynureninase cannot function. So there will be no formation of niacin, so there will be deficiency of niacin and there will be increase in the level of xanthurenate, and that's why vitamin B6 deficiency leads to xanthurenic aciduria and niacin deficiency, which is manifested as various clinical features of pellagra. So along with the niacin deficiency, pellagra like symptoms can also be observed in the vitamin 6 deficiency. Now this vitamin B6 deficiency can also be seen in the patients of tuberculosis who are on the drug isoniazid. This isoniazid is the anti-tubercular drug. This can manifest as vitamin B6 deficiency because this drug can inhibit the enzyme kynureninase, and it also inhibits pyridoxal kinase, and that's why this pyridoxal kinase is inhibited, so it leads to decreased PLP synthesis, and this decreased PLP synthesis is there, and that's why the patients who are on isoniazid, there will be deficiency of vitamin B6, and that's why those patients are always supplemented with the drug pyridoxine along with this isoniazid. PLP also has a role in glycine metabolism. Glycine is converted to serine by serine hydroxymethyl transferase, and there is a tetrahydrofolate is also involved, and it also requires pyridoxal phosphate. What is the role of vitamin B6 in methionine and cysteine metabolism? This is also very important biochemical function. So this homocysteine, it is converted to cystathionine by the action of enzyme cystathionine beta synthase. This also requires serine. Now this cystathionine beta synthase, it is a PLP dependent enzyme. Further, this cystathionine, it is converted to cysteine and homoserine by transsulfuration reaction, and this requires cystathioninase, which is a PLP dependent enzyme. Now what will happen if the PLP is deficient? Then the homocysteine cannot be converted to cystathionine, and cystathionine cannot be further converted to cysteine and homoserine. So there will be increased formation of or increased level of this homocysteine in the blood. So it will lead to homocysteinemia, and that will be excreted through the urine, and it results in the homocysteinuria. As well as if this cystathionase is deficient due to the deficiency of pyridoxal phosphate, then the cystathionine levels can also increase, which will lead to cystathionuria. So that's why deficiency of PLP leads to homocysteinuria and homocysteinemia, and this hyperhomocysteinemia, it is the risk factor for atherosclerosis, and in turn it is associated with the cardiovascular disease, and that's why vitamin B6 deficiency is associated with the cardiovascular diseases. PLP also has a role in glycogen metabolism, that is glycogenolysis by the action of enzyme glycogen phosphorylase, which is PLP containing enzyme. There is release of glucose 1-phosphate and there is formation of limit dextrin. So this pyridoxal phosphate, it is the coenzyme of this enzyme glycogen phosphorylase, and we know that 80% of the pyridoxal phosphate, it is present associated in muscle with the glycogen phosphorylase. The next biochemical function is sphingolipid synthesis, sphingomyelin synthesis. So the reaction palmitoyl-CoA to form 3-keto-spinganine, it requires serine palmitoyl transferase, and this reaction requires pyridoxal phosphate. So if there is vitamin B6 deficiency, there will be decreased sphingolipids formation. So it will lead to decreased sphingomyelin, which is required for the myelination of nerve. So there will be demyelination, and it will result in the neurological symptoms associated with the vitamin B6, that is PLP deficiency. Coenzyme A synthesis is also the important function of PLP. It is synthesized from the pantothenic acid and the formation requires vitamin B6. So we have seen various functions of vitamin B6 owing to its coenzyme form pyridoxal phosphate. Now we will see about the vitamin B6 deficiency. What are the various causes of vitamin B6 deficiency? So the most important cause is isoniazide anti-tuberculosis drug. So this drug will inhibit the enzyme pyridoxal kinase, which is required for pyridoxal phosphate synthesis, and that's how it will manifest as a vitamin B6 deficiency, and that's why along with this isoniazide always pyridoxine is given to the tuberculosis patient. Then women who are on the oral contraceptive pills, it will also result in the vitamin B6 deficiency. The penicillamine, which is used to treat the Wilson disease, can lead to vitamin B6 deficiency. The newborn infants who are fed on the formula milk with low vitamin B6 level can also develop vitamin B6 deficiency. And alcoholics, it is prevalent in the alcoholics as well. Now coming to various deficiency manifestations seen if there is vitamin B6 deficiency. So those deficiency manifestations can be neurological like depression, irritability, nervousness, mental confusion, peripheral neuropathy. So all the neurological manifestations can be seen in vitamin B6 deficiency. Along with that, there can be dermatological manifestations, there can be hematological manifestations. Now what is the biochemical basis for these neurological manifestations? We already know that the pyridoxal phosphate is required for formation of GABA, it is required for sphingolipid synthesis, it is required for epinephrine, norepinephrine, serotonin synthesis. So if the neurotransmitter level is decreased and if the sphingomyelin level is decreased, it results in the demyelination of nerves. So that's why it can result in the peripheral neuritis, and decreased level of GABA, decreased level of epinephrine, norepinephrine, serotonin, it can result in various neurological manifestations. Dermatological manifestations because of the decreased niacin synthesis, it can result in pellagra and there can be irregular indentations in nails. The hematological manifestations because it is required for the first important step of heme synthesis, that is formation of delta aminolevulinic acid, and if it is deficient then it can manifest as hypochromic microcytic anemia because there is decreased heme synthesis. But this anemia, it is the pyridoxine responsive. Pyridoxine can correct this anemia. This is also called as sideroblastic and iron refractory anemia. So this is the picture of peripheral smear showing the sideroblast. So it is manifested as a sideroblastic anemia. Which are various pyridoxine responsive disorders? Sideroblastic anemia. So vitamin B6 deficiency leads to sideroblastic anemia, and if the person is treated, then it can be, if the person is given pyridoxine, the anemia can be treated. Homocystinuria, because of PLP deficiency, it can result in increased homocysteine level, and increased homocysteine level in the blood, it will manifest as excretion of homocysteine through the urine, and it is the homocystinuria. Likewise, also cystathioninuria due to PLP deficiency, xanthurenic aciduria, because now in the tryptophan metabolism the pathway will be diverted to form xanthurenate, and that's why it will be manifested as a xanthurenic aciduria. So all these four manifestations which occur due to vitamin B6 deficiency, they are responsive to pyridoxine. What are the various therapeutic uses of pyridoxine? So it can be used in treatment of morning sickness, to relieve premenstrual syndrome, in the radiation sickness, various muscular dystrophy, and treatment of oxaluria and recurrent oxalate stones of kidney. Vitamin B6 toxicity. Vitamin B6 is the only water soluble vitamin with significant toxicity, and if it is used excessively, it can lead to sensory neuropathy, and more than 200 mg per day will lead to neurological damage. Laboratory assessment of vitamin B6 can be done by direct measure or functional measures. So direct measures will include plasma or erythrocyte vitamin concentration, that is vitamin B6 concentration, and estimation of urinary 4-pyridoxic acid, and this can be done by high performance liquid chromatography. The functional measure, the activity and activation coefficient of red cell aminotransferases, that is aspartate aminotransferase and alanine aminotransferases, can be measured, and the tryptophan load metabolite excretion test, methionine load metabolite excretion test can be used to assess vitamin B6 level or vitamin B6 deficiency. So in today's video lecture, we have studied the chemistry, various forms of pyridoxine and their dietary sources, RDA, biochemical functions, causes of deficiency, deficiency manifestations, and laboratory assessment and toxicity of vitamin B6. Now we will see a case study related to it. A 50-year-old male suffering from tuberculosis. So patient is suffering from tuberculosis and he was given anti-tuberculosis drug, that is isoniazid. He was also given multivitamin supplementation along with it, which he forgot to take, which might be pyridoxine, and he is not taking pyridoxine. So after few days he presented with symptoms of peripheral neuropathy. So there are symptoms of peripheral neuritis and pellagra-like syndrome with dermatitis, glossitis and chylosis. So this is because of niacin deficiency, which is manifested in the vitamin B6 deficiency. So basically the case is of vitamin B6, that is pyridoxine deficiency, because in the history the patient is suffering from tuberculosis. He is on the isoniazid and this isoniazid will cause vitamin B6 deficiency because the patient is not taking the multivitamins also and having clinical features of vitamin B6 deficiency along with the pellagra-like symptoms. So this is a case study of pyridoxine deficiency. These are the various questions related to this case study which can be asked. I hope this video on vitamin B6 will be useful to you. If you find the content on this channel helpful, please subscribe to this channel, like and share the videos and help this channel to grow. Thank you for watching and happy learning.